OK. Bonjour, bonjour et bienvenue. C'est un grand plaisir de vous accueillir ici aujourd'hui and to welcome our distinguished guest, Professor Sir Paul Nurse. It is certainly an enormous honor and privilege to have Sir Nurse with us today as part of the Research Institute of the McGill University Health Center monthly Distinguished Professors Lecture Series. And it is especially nice today that over the next 2 days, we will be honoring our very own Dr. Phil Gould, who we all know so well. And through the lectures that Sir Nurse is going to present to us, we will be honoring, as I said, Dr. Gould. And further details of the scientific presentation will be given tomorrow for those of you who can at the Osler Theatre at 4 o'clock at the MGH. And you're all very welcome. So we're here today to learn about what is life, and who better than Sir Paul Nurse to inform us and to educate us. Professor Nurse is a geneticist and a cell biologist who has spent his career studying the genes, the proteins, the enzymes, and the processes that regulate the cell and how cells divide. He has contributed enormously in our understanding and the defining of the elements that characterize life. Sir Paul Nurse is the director of the very famous Francis Crick Research Institute in London. He's the Chancellor of the University of Bristol. He served as the President of the Royal Society in the UK, Chief Executive of Cancer Research UK, and he was the President for 7 years of the Rockefeller University. For his outstanding contributions and his enormous discoveries that have led us to understand how cells are regulated, how they divide, he was awarded the Nobel Prize in Physiology and Medicine in 2001. And he's received numerous very prestigious honors and awards, including the Lasker Award and the Gairdner Award here in Canada. He was knighted in 1999 and made a Companion of Honor in the UK and awarded the Honor of Merit in 2022 for his services to science and medicine in the UK and internationally. He's received many international honors, and we are indeed extremely honored and privileged and certainly delighted to have Sir Paul with us today. He served for many years in the United Kingdom Council of Science and Technology, advising the Prime Minister and the Cabinet, and he was Chief Scientific Advisor for the European Union. Having spent just a very little time with him this morning, one can't help but be in awe of the incredible contributions that he has made, not only to science and to medicine, but in leadership, in politics, in literature, and more. Beyond science and academia, he has many interests. I learned that he's an experienced and avid pilot, a lover of the arts and the theatre, and a keen sportsman who I understand he ran up Montreal early this morning. And I know it was early because I received an email from him at 4:45 this morning. So I guess he must have been running furiously up the mountain. So nurse, thank you for being with us today. And we certainly look forward to learning all about life. Thank you.
Thank you. Going through all the jobs I've had, it sounds as if I can't keep a job, I have to say. It's a real pleasure to be in Montreal, and thank you all for coming. I've been here quite a few times before, and it's always been a very pleasant time, and indeed it is this time. I'm going to talk about one of the most fundamental questions in biology. What is life? In fact, you could say it is the fundamental question of biology. It's an easy question to ask, not so easy to answer. And I have to now admit to you right up front, you're not going to get an answer today either. But you will get a direction of travel. So I hope you're happy with a direction of travel. There's an extraordinary diversity of life on our planet, of course. All around us are bacteria, fungi, plants, animals, and of course ourselves, human beings. And the question is, what is it that links all these different life forms together? Now, it's a long time ago, but I think it was this butterfly that you see here that I saw when I was about 12 years old in early spring in London. I was sitting in the garden, and it was really seeing this butterfly that made me first start thinking a bit about this question about what it means to be living. This butterfly, quivering yellow butterfly, flew over the fence, hovered, briefly settled, then a shadow disturbed it, it took flight, and it disappeared over the opposite fence. You'd say nothing very exciting about that. But that intricate, perfectly formed brimstone yellow butterfly, that's what it is. It's one we see in the UK early in spring. What it made me think was it was both utterly different to me and yet somehow familiar as well. So like me, it was obviously alive. It could move, it could sense, it could respond. It was full of purpose. And I found myself wondering, what does it really mean to be alive? And that is, of course, the question, what is life? Now, it has to be said, I'm not the first person to ask that question. It's been asked over the years, even the centuries. And for example, this is the title book, the title page of a book, which influenced me quite a lot. It is also called What is Life? In fact, I stole the title of this talk from this particular book. It was written in 1944 by the famous physicist Erwin Schrödinger. It's the only thing he wrote about biology. It was influential, particularly for me, when I first read it over half a century ago. And my approach to this is rather different to Schrödinger's book, although I will quote from it a little bit later. I thought what would be helpful would be to consider some of the ideas of biology and see whether we could pull those ideas together to get a sense of what life is. And really the sort of principles defining the fundamentals of life. And that's what I'm going to do this evening. I'm going to describe 5 ideas, where they come from, what they are, and then pull them together to establish these principles. Now, the first of these ideas is the concept of the cell. Now, the cell is the basic structural and functional unit of life. As I say up here, it is life's atom. They were discovered by Robert Hooke in the 1660s in the city I now live in, Oxford. And he discovered them with the use of a microscope. Microscope a bit like this one I've shown here. We don't have his original microscope. And what he used is he used a razor to cut slithers of cork and then examined them under a microscope looking like this. And what he saw were neat arrays of boxes. And the image you see, the black and white one, is the image that he published, this drawing, in a book published in 1665 called Micrographia. And he called these boxes cells after the Latin word cella, a small cubicle. It was used particularly for monk cells in a monastery. Then, and I put up here a scanning electron microscope picture to show, actually, it's pretty much the same. So 350 years of technology hasn't changed very much what we sort of see here. Different colored this time, of course. A few years later, a Dutch draper, not a scientist, a Dutch draper called Leeuwenhoek, Anthony von Leeuwenhoek, was working in Delft. And he scraped between his teeth and put what he had there under a microscope and discovered single-celled life. He discovered bacteria. This is a picture that he drew at the time. He wrote about 1,000 letters, and he wrote them to the Royal Society in London. This was about 1680. And they're actually all in the archives there. And I've picked this particular letter up, actually, this one here. And he drew what he saw here. He called them animalcules, bacteria. I think what's particularly charming is Fig B, where obviously the bacteria was swimming about under the microscope and did a little loop, as you can see that. Has to be said, he was rather disturbed by this discovery of microbes living between his teeth, because he was very proud of his dental hygiene. And discovering life in your teeth is probably a problem there. And he mentioned this in his letter. Now, he was a close neighbor in Delft to the painter Vermeer, who many of you may be familiar with. And Vermeer was unusual, actually unusual for any painter of any time, but particularly in the 17th century, because he painted 2 pictures, and he only painted less than 40 in his entire career. 2 pictures of a scientist, very unexpected. This is one of them. One is in Frankfurt, the other, this one, and the other is in the Louvre. Now, he turns out to Vermeer also lived in Delft. And I like to think this is Leeuwenhoek. But I have to admit to you, there's not the slightest bit of evidence for this speculation, except when Vermeer died, Leeuwenhoek was the executor of his will. So they must have known each other. So let's imagine we're looking at Leeuwenhoek here. Now, over the next 2 centuries, it became clear that all living organisms consist either of a single-celled life, like bacteria, or a collection of cells, like us. And this conclusion was summarized by a German zoologist, Theodor Schwamm, who in 1839, in German, but it's translated here, we have seen that all organisms are composed of essentially like parts, namely of cells. Did I just say 1838? I obviously got it wrong, 1839. And this identifies cells as being the basic structural unit of life. But they're also, and I'm emphasizing this, the basic functional unit of life, slightly different. And this was indicated by Rudolf Virchow, who is the founder of pathology, another German scientist, both rather serious-looking gentlemen, it has to be said. And he published in 1858, again in German, every animal appears as a sum of vital units, each of which bears in itself the complete characteristics of life. Great quote, a quote that indicates it's a functional unit, the basic functional unit of life. He also described that all cells come from cells. I've spelt form instead of from. I've just noticed there. Okay, all cells come from cells. Omnis cellula acellular. That was quite important because at the time, he wasn't the first to say that, by the way, but at the time it wasn't quite clear whether cells arose by some sort of spontaneous generation or through the division of pre-existing cells. And it's from the division of pre-existing cells that, of course, we know is the case. And I'm going to refer back to that again towards the end of my talk. So cells are interesting, but if I fail to convince you of that, let me show you this picture here. This is a picture of a mammalian egg being fertilized by sperm, and remind all of you, you all once looked like this. Every one of you was once a single cell. And if I fail to interest you now, there is a door over there. I think this is really quite exciting, I think. Now, cells we can consider to be the simplest entities which unambiguously exhibit the characteristics of life. I say unambiguously because I will have to talk a little bit about viruses, of course, as everybody does tackling this problem. But this means that understanding cells is very informative, in fact, key to understanding life. So they will feature prominently in what I have to say. So here is a modern artificially colored EM of a cell. What are they? They are bounded physical entities. So they're separated from their environment by a semi-permeable membrane, lipid membrane. But although separated, they're in communication with that environment. This separation allows order to be built up inside cells at the expense of increasing disorder outside cells. I want to emphasize that because in this way, life doesn't contravene the second law of thermodynamics. And that's a law that often troubles physicists when they think about life, because they see this entity here is confounding the second law. But actually, it isn't. Although it even worried Schrödinger. And this is the top sentence here, an organism's astonishing gift of controlling a stream of order on itself and thus escaping the decay into atomic chaos. Rather colourful, impressive language, I would say, but he was struck by that. I've also put a second quote from his book, which will introduce the second idea I want to talk about you, which was these chromosomes. He'd been talking about chromosomes contain some kind of code script, And in that, the entire pattern of the individual's future development and functioning. And this, of course, is the idea of the gene. So that's my second one, explaining the mysterious phenomenon of inheritance. Now, this is the idea of the gene is the basis of heredity. And it had its origins with the work of the Augustinian monk, Gregor Mendel, who was abbot of Brno Monastery. And he published 2 papers in 1866. Here's Gregor when he was abbot. Now, he tackled the problems of inheritance by carrying out crosses of pea plants with different characteristics and analyzing the outcomes of thousands of progeny formed in these crosses. He was very quantitative, and in this way, he perhaps differed somewhat from biologists, naturalists in the middle of the 19th century that tended to describe, but not actually to be so quantitative. Darwin would be an example of that, and I'll mention him in a moment in this context. But Mendel was trained as a physicist in the University of Vienna, and I think brought a quantitative view to it. Mind you, he wasn't very good at exams and never actually graduated in physics. The Augustinian order was actually a teaching order, but he failed his exams. Just in case there's any of you in this room who have failed your exams. I failed my exams too, but we'll pass over that quickly. Anyway, I visited the monastery in 1981 in the middle of the Cold War. This is what it looked like. And this was the garden in which he did his crosses. And I put it here because it's extremely large, really, in a monastery. And he had a greenhouse of a similar size. And this monastery was essentially a research institute. It had about 8 monks in it, and they all had scientific research projects. I was astonished to learn all of this, and it's something we can't even imagine today. But here, the laws of genetics were discovered in this garden here. And what Mendel did was crossed peas of varied characteristics, including plants of different heights, different flower colors, different seed shapes. Here's some examples of peas and how they can vary. And famously observed in the progeny produced between crosses between these different characteristics of these plants. He counted these characteristics in the progeny and discovered very simple ratios that we all learnt at school, like 3:1 and 9:3 to 3:1 and 1:2:1. I see some of you are nodding, so we must have had the same lessons at school. And this eventually led to what we now call Mendelism. That is the idea that heredity is based in certainly higher organisms, is based on pairs of indivisible particles or factors, as Mendel called them, one inherited from each of the 2 germ cells, pollen and ovules in plants, sperm and eggs in animals. These factors were subsequently found to be linked to chromosomes. But it is important to realize Mendel didn't quite invent Mendelism. Mendelism took a little longer to develop, about another 30 years. And in fact, there's some things about Mendel's work which means that he probably didn't quite get it. For those who are into it, you know, the little A's and the big A's and B's, the way he described it is A, little a, then, AA, little 2 times AA, and then 2 little AA. And he didn't get it was more sort of symbolic for him than exactly as we described. However, Mendelism was formed and named after him 30 years later, when 3 other botanists repeated these experiments, actually not knowing that Mendel had done it all before. It reveals something about the characters of scientists. The first one hardly mentioned Mendel. The second one, who was beaten by the first one, mentioned Mendel a lot, so that the first one wouldn't get any credit for it. And the third one just put up the white flag altogether with that. It is interesting to ask why Mendel no notice was taken of Mendel at the time. And what's important to know is in between the 1860s and the early 1900s, chromosomes were discovered. And here is an example of a late 19th century onion tip of dividing cells showing the chromosomes, and having the physical reality of something separating as cells divide turned the rather abstract arguments of Mendel into a sort of physical reality. And I think it was that combination that allowed Mendelism to really be taken seriously, because it could be seen here. Now, these Mendelian hereditary factors are now, of course, what we call genes. And in the 1940s, Oswald Avery, who was in what was then the Rockefeller Institute, now Rockefeller University in New York, showed that the genes were made of DNA, or deoxyribonucleic acid, not Crick and Watson and co. I'll come to them in a moment. It was actually Avery. And this is the DNA molecule, and you see that you have the chromosome, it's unraveled, and you can see the double helix emerging from that. And the structure of DNA as a double helix was established 10 years later in London by Rosalind Franklin, Ryan Gosling, and Maurice Wilkins, and in Cambridge, who interpreted the data. We won't go into the details of all of that, but we're probably familiar with that. Interpreted the data by Francis Crick and Jim Watson. And Crick and Watson famously argued that the double helical structure of DNA could explain heredity through the linear sequence of the bases making up DNA and the pairing between those complementing bases, adenine pairing with thymine and cytosine with guanine. And that is seen here. The double helix is like a twisted ladder. Imagine a ladder with rungs, and they're made up, of course, of a G with a C and an A and a T, and then confuse it a bit by twisting it into the double helix. So you sort of lose the straightforwardness of that. And that explains how DNA can be copied during cell reproduction, because you can pull those the rungs of the ladder apart and then copy a exactly complementary copy on that particular template, and that produces two identical molecules from one molecule. And it also explains, of course, Schrödinger's code script and the permanence of the gene between generations through this, because DNA acts as the code script, which is precisely copied every time a cell divides. Crick, very imaginative person, went on to propose what he called the central dogma. Typical Crick. I don't know, I have met Crick quite a number of times, and central dogma, sort of exactly how he was, explaining how, but he was often right, by the way, with his central dogma. Anyway, explaining how the DNA encodes the structure of proteins. And the central dogma describes how the sequence of bases, and you can see that on this figure. I've stolen all these figures from textbooks, where none of these things have I drawn myself. Well, maybe there might be one or two here. I'm trying to remember. Anyway, the gene is copied from DNA to ribonucleic acid to RNA, and that acts as the messenger between the DNA in the nucleus, where the chromosomes are, and the cytoplasm where the proteins are made. And there's one gene in principle encoding each protein. These discoveries are amazing because they are the foundations for molecular biology, and we are still making use of these extraordinary advances 70 years later, and are, of course, critical for understanding what life is. Central to this is the genetic code. Heredity is written in a 4-letter code of the 4 bases, adenine, thymine, cytosine, guanine, and the information is stored in a linear form. I'm going to refer to this because, because this way of storing information is the same as the sentences I'm speaking to you. That's a linear form. Words and sentences in a book, or bytes in a computer. It's all storing information in a linear form. And it means the linear digital storage of information was invented by life over 3 billion years before the computer age, just to put us in perspective, 3 billion years. The nature and function of the gene introduces the next two ideas I want to talk to you about: life as chemistry and then life as information. Now, the functioning of living organisms based on chemical activity and physical forces, the origins of this idea can be traced back to Lavoisier, the father of chemistry, who unfortunately lost his head in the French Revolution because he was also a tax collector. And one thing actually I should just remark here, isn't it extraordinary how so many of these basic ideas of biology are hundreds of years old? I mean, we don't often think about that. We think everything was invented in the last 20 years, 30 years. But, you know, from 1660 onwards, some of these fundamental principles are now really, some of them are very, very old indeed. Now, the functioning of living organisms, as I said, can be traced back to Lavoisier, the chemical basis for it. But it was Louis Pasteur who carried out experiments on sugar beet fermentation, which led to more clearly to the idea that life is based on chemistry. This is a rather romantic picture of Louis Pasteur. I think it's in the Pasteur Institute, looking intently at this bottle, okay? And Pasteur was analyzing the fermentation of sugar beet to make alcohol at the request of industrialists in northern France, where he was working, because they were doing fermentations to make alcohol out of the sugar beet extract. And sometimes it went wrong and it made acid. And they called on Louis Pasteur to see whether he could help that industrial problem. This is applied research. I emphasize this because there isn't such a distinction between discovery research and applied research. There are differences, but not as great as people think. And he demonstrated that the successful production of alcohol required the presence of a single-celled living organism, yeast, budding yeast. And he proposed that the yeast cell produced substances that made alcohol from the sugar, which was derived from the sugar beet. And if the yeast was not there, then he postulated that were perhaps bacteria under making the fermentation, and that made acid by mistake. Okay, so he solved the problem for the industrialists. As long as you put yeast in there, you'll get alcohol. Okay, I don't know if they paid him anything for it, but he solved it. But Pasteur, being a brilliant scientist, concluded something more generally. And he concluded that fermentation was a physiological act yielding products for the cell, that chemical reactions are an expression of the life of a cell. In other words, that experiment led to the idea that life is based on chemistry from the 1870s. So it's chemistry, but it's a very elaborate, a very special chemistry. It's based on carbon polymers, which we've met, DNA, RNA. And I now want to consider a new carbon polymer, that is the protein carbon polymer, because the substances from yeast that Pasteur postulated, because he didn't know about this, that made the alcohol are proteins acting as enzymes which catalyze chemical reactions that turn the sugar into alcohol. Now, proteins are polymers of amino acids with a backbone and side chains of amino acids. And this is shown here in this figure. And what I want to emphasize here is the backbone is common. Okay, you can see it there, carbon and nitrogen based, but it's a variety of amino acids here, but these are chemically diverse. So you have lysine, which is basic, hydrophobic ones, leucine, large and complex tyrosine, aspartic acid, which is acidic. So what this means is the rather boring chemistry of DNA is now here turned into a much more varied chemistry in these proteins. And it's because of this variety of chemistry that actually you can now carry out chemical reactions based on protein. And that's this is what's clever about life. You know, you've got a linear digital storage, very stable, and you turn it into these structures, which are chemically diverse. Not only that, the diverse chemistry it means that this protein chain that you see here, which is just to show you the example of it, and this is the chains, what they look like, can fold up into different structures of different shapes, different sizes, and with different positions of amino acids with different chemistries. So we've now got an extraordinary chemical variety that is seen in the protein molecules encoded by those linear DNA molecules that are chemically inert and stable. And there's 20 different amino acids in life, and that can account for the diversity of chemical reactions that enzymes can take place. Again, I want to emphasize this because when you read this in a textbook, it doesn't read like that. And I mean, it's saying the same thing, but it is extraordinary what is happening here. Now, enzymes and other proteins act as chemical machines. They produce a huge range of chemicals that are required for living organisms to function. They make new molecules, they break them down, they recycle them. They can generate large assemblies which can synthesize these polymers of nucleic acid, protein, and others like lipid and carbohydrate. They can also do physical work. They can make motors which carry cargoes around the cell on protein-based trackways. They act in hybrid ways, combining chemistry and physics, capturing energy from sunlight to produce energy-rich chemicals that can drive the other activities. Now, this is, again, well described in the biochemical textbooks, but something else that is extraordinary about this is that all these thousands of different chemical reactions are going on in a tiny little space, which is only micrometers across. And they are all highly diverse and different. A small sample of the sorts of reactions going on the cell are this map of the central metabolism of a cell. And for those aged over 60, they may recognize this from a textbook of the time. And what you see in the middle there is the red is the Krebs cycle at the very bottom. And we all had to learn this stuff when we were students. Do you still have to do it now? Oh. Now we've got Mr. Google. Why do we enforce it on our students? Anyway, that's a talk for another day. So this is whoops, excuse me, turned over too much. But the point I want to emphasize here is all these dots are different chemical reactions, yet they're all going on in a tiny little space. Yet each of them have different chemical microenvironments. Just think about that. Hundreds and hundreds of different chemical microenvironments. So they have to be somewhat separated one from another for this to work. And this is achieved by compartmentation, initially brought about at the lowest level by the surfaces and folds of enzymes, which make little chemical microspaces, or enzymes working together in complexes, or at the level of the cell, schematized here with these different cell compartments, which divides the cell up, and it's not normally described like this, into different sort of chemical compartments, where you can actually undergo all the hugely elaborate chemistry of life, because the structure of the cell is bringing about highly diverse and different chemical microenvironments. Again, we don't think of it like that. We think of it as sort of just a structure and stuff, but it's actually, that's what it's imposing on the cell. Now, however, for all of this to work together, it has to work as a whole, has to be connected. And that means it has to be organized together, which requires communication between all these different environments and compartments. Communication is the transfer of information, and that leads me to the next idea, is life as information. So you have to complement life as chemistry and physics with life is also based on information, on managing complex systems which are controlled and which result in purpose. I continually use the word purpose to agitate you a bit, because biologists sort of get a bit nervous when we come to purpose. So I'm happy to talk about that a little bit later. I got interested in this, so I thought, when did people start first thinking about life as a complex system? And you're going to be astonished by this, because the first thing I found, I mean, sorry, not the first thing I found, took some time to find it. The earliest reference I found was writings by the philosopher Immanuel Kant, which I'm sure surprises all of you. And he wrote in a book on moral philosophy about 20 pages on this topic. I have not the slightest idea why, but he was very fascinated by complex systems in the base of life. Now, I want to give two examples of information. One I've already explained to you. Now, the structure of DNA is double helix stuff. It's iconic, it's beautiful. Although I did say in the Crick Institute, the last thing I want in there is yet another artistic sculpture of the double helix. I'm just sick of them. You go to any institute and you find a bloody double helix somewhere, you know? And so I've banned it. It's crept in. It's crept in because I wasn't looking all the time, but I really didn't want it. Now, the structure of DNA, however, is beautiful, but its real beauty lies in the fact that it only makes sense when it is recognized as a digital information storage device. I mean, that's what's really beautiful about it. One written in this linear script of 4 letters. And the linear polymer of DNA, as I've already hinted, is ideal for storing information. All the encoding bases are turned inside, and they can get protection, and it's stable, and it's chemically fairly inert. But the information, again, if I've hinted at this, can be turned into chemical action to do work through the varied activities of the proteins which are based on that structure. So you have this linear storage device in chemistry derived from it. For me, this is a wonderful characteristic of life. And until I was thinking about this, it never really fully occurred to me. And it's all built on polymers, carbon-based polymers, the ability to encode information in inert, one-dimensional linear structure, but to carry out chemical and physical work in 3-dimensional chemical structures, which are chemically varied. So that's my first example. The second one I want to tell you about is regulation, using information to regulate. That is making the complex systems in living organisms to act as a whole with purpose, to achieve something, to grow, to be homeostatic, to reproduce. A familiar example of regulation is found in man-made machines. And this is an example I want to tell you about, is a governor. Governor, people think it was invented by James Watt, who sort of optimized the steam engine. It was actually, I think, Leibniz or somebody who did it for regulating windmills. But anyway, it was hijacked by James Watt to regulate steam engine. I took this picture in New Zealand in an 1890s boat, which had one of these things doing it. And I don't know if you're like me, I understand most machines built before 1900 and increasingly none as the century go on after 1900. So I tend to keep my metaphors and analogies to ancient things, because they're sort of understandable. Because look, what happens here? When the machine goes faster, those balls swing out, okay? They close off a valve, shut off the steam, and so it slows down, and then they go back in again and the valve opens, okay? So this is an example of homeostasis, keeps a constant speed of the steam engine. And it's an example of a negative feedback loop. Drawn here, I drew this one, you can see it's crude. A negative feedback where A goes to B to C, the green arrows are enzymatic reactions, C inhibits A to B, so as C builds up, it inhibits A to B, and so C drops. Very simple. Positive feedback, completely the opposite, A to B to C. As C builds up, it promotes A to B, so it's an irreversible switch. These sorts of controls in biological systems were really first properly discussed by the French geneticists Francois Jacob and Jacques Monod. Brilliant analysis of just geneticists, just counting colonies on plates came up with this stuff. I mean, in the 1950s and 1960s, but they were studying gene regulation of sugar metabolism in Escherichia coli. Now, many such feedbacks and control modules operate in cells, maintaining homeostasis like this negative feedback loop, generating switches like the positive feedback loop, but also generating timers, toggles, oscillators, turning and translating that chemistry into what I would call informational modules. So we're now going another step from the chemistry into informational modules. These can be linked together to generate even more elaborate routines of informational management and processing. Now, a metaphor for all of this is a electronic circuit of linked modules. I stole this from Embo Journal, I think, and some wit took a transistor radio and covered it with oncogenes here, which are all sort of connected in this sort of way. Now, one thing you'll note about this, these are all hardwired together. But an even better metaphor for living systems is wetware. That was coined by Dennis Bray, if I remember correctly, which, and what he meant by wetware as against hardware is that because you're connecting the chemistry through water, you can actually rewire how things connect. And that's something, again, that we haven't sort of properly sort of thought about, because you can connect different components in it. And that can lead to actually significantly more complexity in how these systems work. And I think over the years in the future, we'll see more of this. Now, the management of life's chemistry by information brings about allowing the behavior of a complex system ultimately to act as a whole with overall purpose. Do we understand that yet? No, we do not understand it yet, but it is what we need to actually understand. And the cell is the first place, I think, where that's going to happen. I've had several conversations today about the importance of physiology and understanding an organism. And this is the same point, except this is within a single cell, because that is probably where we'll crack it first, if you ask me. And these overall purpose, they can reproduce a cell, make a butterfly, maintain our cells. Now, for centuries, the generation of such purposeful behaviors was thought to require a designer, a divine creator often. And it took evolution by natural selection to change this. And this is my final idea. Evolution by natural selection, how living things came about. Now, this is a beautiful idea. It's simple, but beautiful. Mainly due, but not entirely due, to Charles Darwin. It has two parts. The first is life evolves over time. The second is a major mechanism for that evolution is natural selection. Two separate ideas. The first of them absolutely was not Charles Darwin's idea. It had been discussed for certainly over half a century before. And I have I want to show you here Charles Darwin, the rather serious Victorian one on the right, and his grandfather on the left, Erasmus Darwin. Erasmus Darwin, together with others like Jean-Baptiste Lamarck, speculated quite a lot about evolution. Erasmus was a colourful character, a scientist, a doctor, a poet. He wrote all his research up in poems. It's all in verse. I've got a collection of his books, all published in the late 18th century, all written in poems. He was a Republican. He refused to be George III's physician several times because he was a famous doctor. He was a proponent of women's education and wrote a textbook on what women should be taught. And he was a great conversationalist, despite having a terrible stammer. And he did all his work and writing. He was a member of the Lunar Society that some of you may have read about in the Midlands of England, and there wasn't a university in sight. No, miles away. Anyway, He and Charles, and I'll come to Charles in a moment, discussed the evolution of life, and that implies there's a tree of life, that living organisms are all related. If life only arose once and survived once, as seems likely given the conserved chemistry and coding that we have from bacteria to our cells, then all life on Earth is related to each other. That's something else that's quite profound, really. This connection of life was captured in the only diagram in Darwin's Origin of Species, published in 1859. This is the tree of life. It's the only picture in that book. Notice that Darwin wasn't sure whether life arose more than once. If you look at the bottom there, from A to L, underneath, he didn't know if it's once or more times. We don't know that either, actually. What we probably can conclude is that even though it might have arisen multiply, what survived is only one, which is the one that we have today. We might want to talk about that. By the way, I have no good ideas. Well, I have a few ideas about the origin of life, but no, it's quite a mystery, I have to say. Now, Charles Darwin gathered evidence in favor of the evolution of life through natural selection with his famous trip on the Beagle going around the world. Briefly, what is natural selection? It's the consequence of populations of a species exhibiting variations that are caused by inheritable genetic changes. These can be caused by mistakes when the DNA is copied, or if the DNA is damaged by external causes. Darwin, of course, had no idea about DNA, but this is what he was postulating. If these variants influence characteristics that make an organism more successful, therefore better adapted, and therefore able to reproduce more, then that particular genetic variation will spread through the population. Simple, brilliant idea. Over time, such genetic variations accumulate and lead to changes in the population such that the individuals of that new population will no longer be able to breed with the original population. And that's why the book is called The Origin of Species, because that's what actually was interesting him. A new species which is better adapted and more successful and survives better and reproduces better. And Darwin came to this view in part by his studies of finches on the Galapagos Islands. And you see the finches on the left here. The Galapagos Islands are volcanic islands. They were colonized in relatively recent times, millions of years, but not tens of millions of years. And you'll see the one at the top there is probably cracks nuts, and the one at the bottom probably is catching insects under bark. Okay, so you can see that. They become adapted to their lifestyle and environment by being specialized better for what they had to do to succeed. Just like the pliers on the right are specialized to do different jobs. But the difference between beaks and pliers are that pliers are intelligently designed by human beings for different specialized purposes, whilst the finch's beaks achieve specialized purposes by natural selection without a designer. And that just sort of emphasizes the relationship between these two things, but actually their differences. Now, for natural selection to take place, three things are needed. You need something to reproduce, clearly. It needs to have a heredity system, clearly, and that heredity system needs to exhibit variability upon which selection can work. And this is based on the ideas of the cell and the gene, which I've actually introduced to you. I want you to imagine a single-celled life form with a brown cell coat. That looks orange here, but anyway. The color of which is determined by a gene. Imagine when the cell copies its DNA in that first division up there, it makes a mistake. So the cell coat gene is altered. And the cell now makes a red coat. You see that down at the bottom right, yeah? As a consequence, also imagine that red-coated cells survive better. Perhaps they avoid predators. It doesn't matter what it is. And as a consequence, the red cells replace the brown cells in the population, and whizzo, evolution by natural selection has taken place. This is a profoundly important idea in biology, so much so that the 20th century geneticist Hermann Müller used it to define what life is. He defined life as living things have properties which allow them to undergo natural selection to evolve. Now, before I consider this further, and I'm getting near the end of my talk if you're getting tired, so deep breaths, deep breaths. I want to say goodbye to Darwin as an old man. This is what he looked like a bit later. And quote his final monumental sentence in The Origin of Species. Whilst this planet is gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Why can't we write papers like that anymore? By the time we've submitted a paper to a journal and it's gone through the reviewers, the editors have deleted anything of any interest. I can't even read my own papers a year after they're published. And yet I can read this stuff nearly 200 years after it's done. We have lost something. We've lost something. Get rid of reviewers of papers probably is the best thing. I want you to note he was relating his work to the great physicist Isaac Newton, referring to gravity. He was making a point there. He was saying biology can have fundamental laws just like physics. And it's not accidental that he mentioned gravity. It's subtle, but I'm sure that's what he meant. Now, I'm about to say what I think we have to think about with life. I'm going to bring it together. But before I do that, I have to talk about the difficult category of intermediate life forms, in particular viruses. You know, this is the O-level question, are viruses alive or not? Okay. Now, viruses have a picture. Here's a virus. It's a bacteriophage, bacterial virus, they have a genome based on DNA, can be RNA. They undergo evolution by natural selection, as we saw with COVID, thus passing Muller's test, but can only reproduce when they are inside the cells of other host living organisms. And they operate by hijacking the cell's molecular machinery to copy the virus's genome and make more viruses. This means that a virus is completely dependent upon another living entity. You could also say, of course, that a virus is alive when in a cell and may be a lifeless chemical when outside a cell. That's what some people have argued. So the question is, are they truly alive? Now, in thinking about this, I think it's important to remember that all other forms of life are also, to a greater or lesser extent, dependent on other living beings. And when that question is asked, that isn't often thought about. The many parasites that live on or inside cells or bodies of animals, plants, or fungi are examples of that. The extent of their dependencies are less than in the case of a virus, but it is not a clear distinction, as people sometimes argue. Other organisms are used as food. We are reliant on those other organisms. There are certain amino acids we cannot make, and we have to obtain them from our diet, from other living organisms. So the reality is there's a graded spectrum of living organisms. There are different life forms with varied dependencies on other life forms. In the case of the virus, that dependency is very strong, whilst in other living organisms, the dependency can be quite weak. But it isn't a complete black and white situation. And that, I think, helps us clarify the situation in thinking about viruses. It's extreme, of course, but we're all highly interdependent. Therefore, this is a graded spectrum, but because these different life forms evolve by natural selection, I'd argue that we should consider them alive. It also means something else, that life on Earth is fundamentally connected in two ways, both through being related as a result of evolution and the tree of life, and also through these deep relationships and interdependencies. So I think I'm going to mention that finally at the end, so I'll come back to that. Right. I'm now going to say what I think we should think about, the direction of travel, as I said, about what we need to think about the principles of understanding what life is. I want to warn you, it isn't leading to a neat dictionary-like definition of life. What is life? They are independent, bounded physical entities. Basic unit of life on Earth is the cell. Separated from their environment in communication, they can generate order and organization. We and other multicellular organisms are aggregates of cells. Living things, and it's the key slide, possess the ability, I got this from Muller, possess the ability to evolve by natural selection. And to do that, they have to grow and reproduce and have a hereditary system which exhibits variability upon which selection can act. Living things are chemical, physical, and informational machines. They construct their own metabolism. They use the metabolism to maintain themselves, grow, and reproduce. These are all the things we have to hold in our head. And it's very descriptive, I know, but we have to hold it. When we're thinking about life on this planet, life on Earth is based on carbon polymer chemistry. The polymers produce nucleic acid, based linear information storage devices, that term again. The protein is based on chemical, leading to protein-based chemical and physical machines. Integration of all these functions requires the management of information. Inputs are gathered from within and outside the cell, processed, stored, and instruct cells and organisms to maintain themselves, to grow, and to reproduce. Living things on Earth are related by common descent and are closely interconnected and dependent upon other living things. Some of these characteristics may not be found in other life forms, but are found on Earth. Should we ever encounter life elsewhere in the universe, and probabilistically, the Drake constant and so on, they say equation, that seems likely. We may never find it, but the universe is so big. I suspect it will be built on rather similar fundamental principles. The chemical details may be different. It may not be based on carbon even, but to satisfy the need for long-term information storage, I think the chemistry will be built on polymers, because the polymers are the linear storage devices, and we've seen how life turns it into chemistry action. However, the only corner of the universe where we know for certain life exists is here. The life that we are part of here on Earth has bewildering diversity. We're beginning to make some sense of it, and it makes an understanding a fundamental contribution to our culture and civilization. I think the ideas that I've talked about today they contribute to our civilization, our culture. Our growing understanding of life also has great potential to improve the lot of humankind, as is obvious to in a medical audience. But the knowledge goes further. Biology shows us that all living organisms we know of are related and closely interacting. We are bound by deep connectedness to all life. To infecting bacteria, fermenting yeast, pea plants, and this yellow butterfly, who have accompanied, of course, these on our talk, my talk. Now, together, all these different species of life are life's great survivors. They're the ones that have actually got through natural selection to where we are. And these are the latest descendants of a single, immeasurably vast family lineage that stretches back through an unbroken chain of cell divisions into the far reaches of deep time. And that's something I hadn't fully recognized either. We are connected by repeated cell divisions, which bridge the generations and make us, that go back to the beginning of life. It's something really quite extraordinary. However, as far as we know, we are the only life forms who can see this deep connectivity and reflect on what on earth it might mean. And I argue as a consequence that that gives us human beings a special responsibility for life on this planet, made up as it is by our relatives, some close, some more distant. We need to care about it, We need to care for it, and to do that, we need to understand it, which is why I've given this talk and actually also written a book, which is covers the same material. Thank you very much for listening to me.
Thank you so much for inspiring us, for teaching us, for giving us much to think about. Sir Paul, it's been an absolute honor and a privilege. Would you mind if we had one or two little questions?
I'm happy to have any questions, but I'm a bit deaf, so you may need to tell me the question.
So do we have any burning questions? I know we're beyond the hour and we will have time to network around some cocktails, but if you do have questions, we'll...
And if anybody wants to leave. I'm not going to be insulted by that. That is fine. They don't have to be burning questions, just questions. Okay.
Questions. Do we have any questions from the audience? Yes, Nada, please, Dr. Jabada.
I don't have a question. I just wanted to thank you deeply for your presentation. I think it's a privilege to put things into perspective. We forget our beginnings. We forget how you said about science being about 100 years ago, mathematics were even before, and we still are living by the same theorems that people with much less instrument that we have today have. We should be more humble, and I think we should reflect way more on what the meaning, on what are the order and the magnitude, instead of just looking in silo. And just wanted to thank you for showing us that again.
Well, thank you for your nice comments. I do like to explain where these ideas come from, because we forget, and they're profound and they're quite old.
Yes, Dr. Barillet.
Yeah. Okay, so origins of life. I mean, what is it? Let's just say how extraordinary that is. The solar system formed 4.6 billion years ago, roughly, and that the Earth cooled down probably to a low enough temperature. They guess a bit at this by 4.2 billion years ago. The first fossils 3.5 billion years ago. So in a period of 500 million years, we evolved life, which we can see as a fossil. It took another 2.5 billion years to make multicellular life. Yet I'd have thought making the cell was more difficult. So it didn't take so long. I mean, it took 500 million years, which of course is quite a long time. But it is something that we need to wonder about. And people like Fred Hoyle just imagine that, well, it came in on a comet from somewhere else, which is a bit of a cop out, because it's still got to have risen somewhere. I think I would start with RNA. RNA has the ability to both encode information and also do enzymatic reactions. And I mean, obviously not my idea. It's been around for 30 years or 40 years or even longer. But I would start probably there. I'd probably start also thinking about vents and little compartments in rocks which might act as initial cells somehow. So you don't have to invent everything at the same time. But I don't really have a good explanation that I can believe. The one you were probably thinking about was maybe the clay particles, and that is a very clever idea. The idea is that clay particles can evolve by natural selection, by flaking off in certain directions, and therefore seeding more clay particles, and that somehow could be the base upon which then more complex life could form. Is Cairns, I think, Smith or some, I forget actually who wrote it. And it's a clever idea, but I'm not terribly think it's likely to be true. But the reality is it's a mystery. It is a mystery, because it is difficult looking back 3.5 billion years and knowing what's going on. So I put up the white flag. Once we have a life form of the sort, I'm happy we can produce anything, okay? But getting that initial thing, tricky stuff.
Did you hear the question? How a nucleotide's made?
Ah, yeah. By the way, thank you for nodding so vigorously during my talk. It was very encouraging, if I may say. Making the that's I mean, we could make probably nucleotides could be made chemically before they have to be made enzymatically. So you can sort of imagine scenarios, but you've got to put all these different things together. You've got to put a cell together, a gene together, and the coding together. And it only works when it's all together. So it's got to be a stepwise business. But I think we could make the building blocks of life with concentrated chemical constituent chemicals and lightning and stuff. I mean, all this sort of thing. So we could imagine it, but it's getting it all together at the same time. That's why I want little holes in rocks to sort of cope with cells, RNA which can do both encode and also do enzymatic reactions. One over there. You want to get to the cocktail?
Oh, yeah. Thank you for the wonderful talk and for having the time to take questions. I wanted to ask if you would consider that death should be part of the definition of life. How would you fit it?
Absolutely the case. Without death, we are not alive, which sounds incongruous, but of course, for evolution by natural selection to take place, you have to have successive generations. And successive generations are brought about by death. If we were all immortal, evolution would stop.
Thank you.
I didn't mention it, but you're completely right. So perhaps we should be fonder of death. Not sure I am though, but I just thought so. One over here. I'm sorry.
Yes, thank you for this very thoughtful lecture. So I was wondering if you thought that biological systems actually had a way of recording the need for change. So purposeful evolution as opposed to natural selection.
Well, I used to ponder quite a lot about that question because the difficulty of that is it's sort of predicting the future, and so how could, in a very general sense, let's give an example. When when DNA is copied, there is it's not copied precisely, it's deliberately copied slightly inaccurately. We could make DNA polymerases that copy the DNA so they're more accurate. They are not as accurate as they could be. And that might be a mechanism to introduce variability upon which future natural selection can work. Because if you're too stable, you may be perfectly adapted for that environment at that point in time with other living things around you, but you're not sufficiently flexible and varied and therefore flexible to evolve. So people have written articles about how much variability there should be that can lead to that, but it is very theoretical and I've never really fully understood it, so I've not done it. But the point you're making is something we do need to think about. Yes.
Oh, hi. With regards to life needing to be connected and depending on each other, what are your thoughts on the selfish gene and the other works of Richard Dawkins?
Well, the selfish gene, I mean, it's useful to think about that. It makes a very gene-centric picture. The unit of selection is actually above the gene. In single-celled life, it could be a single cell. So you do have to encompass the entity which is being selected. I mean, we say the gene is selected, in that sense it is, but it's the phenotype that's selected that has consequences on the gene. I think Richard sort of gets that, but I think he likes the term the selfish gene because it sells books probably. But I mean, so that's one thing. And then, of course, we have in societies of animals interacting with plants and so on, you get much more complex interactions taking place where you may have group selection occurring, which again can have a genetic origin, but it's complex to work out. So the evolutionary biologists talk a lot about this, and as I just said from over there, I get a bit lost in it, but I don't think it's a fundamental problem. The selfish gene is just a more extreme version of it. Nothing wrong with it, but it may throw away some of the interesting things upon which the gene has to work, because you don't bother to think about it. And you need to think about the organism as a complex system to understand how it all works. If I can add something about it, I get a bit tired of the arguments, you know, of holism versus reductionism. You know, so you get these books published and saying, you know, genes are all wrong and stuff. I mean, actually, it's both. And it's so obviously both. I don't know why people get so hot under the collar about this. We have to be both reductionist and holistic. I know you didn't ask that question, but I'm sorry.
We will have the last.
Our honoured guest here.
Oh, no, I think that the only problem with this lecture is it's coming at the wrong time.
The wrong time.
It should be the first lecture given to any student in biology. And from that point, you're going to have to travel much further and much faster. Thank you so much.
Thank you. Well, not at all.
So in closing, I really want to acknowledge, as we move on to our cocktails, I really do want to acknowledge the support that we have got from the Montreal General Hospital Foundation for the support. And especially I want to thank Stephanie. Perhaps you'd like to come up and say a few words, Stephanie. Stephanie and her team have been incredibly helpful and supportive in facilitating and enabling this wonderful opportunity that we've had today and that we will continue to have tomorrow. So, Stephanie.
I am standing between you and the cocktails, but it was a wonderful afternoon and a privilege to spend the afternoon with you. And thank you everybody for coming. And I would just like to make the link to our support to this wonderful lectureship series that has been organized by Dr. Taus and her team and the Research Institute with Dr. Phil Gold, who's here with his wife, Evelyn. And this month and next month marks a very important celebration that we have been working on together for some time. The research building at the Montreal General Hospital site will be named officially in Dr. Gold's honor. And that will be opened on June 5th. And the other part of the celebration is kicked off this afternoon, and then tomorrow the inaugural lectureship in your name, Dr. Gold, the inaugural Phil Gold Distinguished Lectureship. And I can't think of a finer name and a finer person to give it. And so we're all very proud to be here supporting it and to make these wonderful links. So much of your speech, I think, resonated with why we're all here and why we're involved. So thank you very much. Merci. Bon après-midi.
So please join us with a cocktail, a snack, and for some networking. Thank you.